you have any question until now about the topics of today? No? Everything is clear? So, I talked about again, as a reminder, we need millions of BCRs or TCRs. Okay, Haji? Mr. Haji? Here. We need millions of TCRs or BCRs. But we have just thousands of genes, 20,000 or at the maximum 30,000 of genes. So this amount of genes cannot produce normally 10 million BCRs. We need some processes that enable just this, the process that I'm going to talk about this process just is active in B cell and T cell. And this process enables the B cell and T cell to produce millions of TCR and BCR from thousands of genes. And these 20,000 genes or 30,000 of genes, just few of them are for BCR and TCR. Rest of them are for the rest of the proteins of the body. The total genes of the body is 20,000. And from these 20,000 genes, just few of them are for TCR and BCR production. The rest of them are for the rest of the proteins of the body. Many enzymes, many proteins are produced in the body routinely. You know that, for example, this is a DNA sequence, a gene on the DNA, a small piece of DNA on one chromosome. This is a small piece of DNA on one chromosome, or indeed a gene, a specific gene. And we have RNA polymerase that it produces, what is this? Making of RNA from DNA. What does that mean? Transcription. And this is an immature RNA. Okay? From a gene by transcription with RNA polymerase, it doesn't matter the name of the enzyme. We will have immature mRNA. This is called mRNA or any kind of RNA. Routinely it is called messenger RNA. Do you remember?
Yes.
So this is immature. Needs some changes to become a mature RNA. So you know that on the immature RNA we have exons and introns. Do you remember? Introns, these parts have to be eliminated from the immature RNA, and the exons bind to each other. This process is called splicing. Okay? So DNA, transcription of RNA from DNA, and we have an immature RNA that it has both introns and exons. We need just exons, and introns have to be eliminated from the immature RNA. Now this is a mature RNA. Okay? And from a mature RNA, we will have translation. What does translation mean? The RNA, the sequence of RNA into amino acids. Production of protein from mRNA. Translating the content of mRNA to amino acids or making protein. So you know this. We have transcription, making of RNA from DNA, and then translation, making protein from RNA. They are the basics of biology. So, but sometimes we have a process which is called alternative splicing. Alternative splicing means that, for example, if there is 4 exons on the immature mRNA, in each version you can see we have 3 versions of mature RNA from the single mRNA, immature RNA. Again, we have 3 different versions of mature RNA from just one immature RNA. This one has all exons. This one just has 1, 3, and 4. The 2 is eliminated from this one. And this one has 1, 2, and 4. 3 is eliminated this one. This process making different versions of mature RNA from one single immature RNA is called alternative splicing. Please, tonight, all of you, this is an assignment for you. All of you need to study about translation. Write it down, please. Write it down. You need to study about transcription. I do not need to study the details of the process of transcription. It's important to know what transcription is. So you need to study transcription, translation. This is an assignment for tomorrow. Transcription, translation, the difference between immature RNA and mature RNA, just mRNA, messenger RNA, not other types of RNA. And splicing, the difference between splicing and alternative splicing. Okay? You need to study tonight. No question. This is a DNA, a gene on the DNA. Haji, have a look here. A gene on the DNA. That's one, please. Have a look here. Please. We have a gene on the DNA. Making of immature RNA from the DNA, which is called transcription, and on the immature DNA we have several exons and several introns. And introns, all of them have to be eliminated by splicing. And from one mature RNA with different exons, we can make different versions of mature RNA by elimination some exons. Okay, this is called alternative splicing. It enables the body to produce 3 different proteins at least from just one gene. Okay, each of these mature mRNA, all of them are different from 1, 3, 4, 1, 2, 3. So some parts are absent in this. From some part of this one is absent in those ones, and they are different from each other. So if a translation of them to protein will produce 3 different proteins. Okay? Is that clear for you? Another important basics of biology is that sometimes a gene is a complete gene. From the beginning to the end is the sequence of one gene which has composed of introns and exons. But sometimes this is called continuous gene. So continuous gene is a gene that from the beginning to the end, all of them is the content of the gene. But we know that in all genes, we have both exons and introns. They are part of the gene. But sometimes we have a discontinuous genes. It means we have a gene which is made from different fragments. A gene which is composed of different fragments. They are called discontinuous gene. The genes of immunoglobulin or antibody, they are same. Or BCR, you know they are same. Immunoglobulin, BCR, or antibody are discontinuous gene, and TCR. All of both BCR and TCR genes are discontinuous and fragmented, or fragmented gene. And that's why we can use these fragments to produce, I'll tell you how, different BCRs. So until now, it's important to know that the genes of BCR and TCR is fragmented gene, discontinuous gene. Okay? No question? Clear? And those fragments, before transcription, this is DNA, a DNA sequence. Before transcription, these fragments have to bind to each other, and unwanted DNA have to eliminate it. So the process of binding those fragments together is called recombination or rearrangement. Rearrangement means binding of different fragments of each DNA, each gene, together to make now a complete gene from the beginning to the end. And all of them are on the DNA. We are not talking about RNA. Discontinuous gene, binding of different fragments together, which is called recombination. Another thing that tonight you will need to study is recombination and difference between continuous and discontinuous gene. You need to write your answers and give it to me tomorrow. What is recombination? Just you should answer in 1 or 2 sentences, not more. I do not need the detail of the process of recombination. And the difference between continuous and discontinuous gene. Okay? You need to write the answers on your papers and give it to me tomorrow. And it's a part of your scores for the exam. It's an assignment. It's a part of your final score. Is that clear until now? So the genes of BCR and TCR is a fragmented gene, and those fragments have to be bound to each other to make a continuous gene. And now we will have transcription to make the RNA and the rest of the story. So before making the RNA, we need a process, which means recombination, to make the gene complete. So recombination is a process where pieces of DNA are broken. So those pieces are broken from these parts, and then they can bind to each other with a specific kind of enzymes. All of them are made by enzymes. The cutting of DNA from these parts and binding them together, all of them are made by different types of enzymes. Okay? So antibody genes have multiple fragments. And we have multiple versions for each fragment. It's very important. What does that mean? This is indeed the sequence of the DNA or gene of antibody. This is the sequence of variable region. This is for constant region and the rest of the constant region. So we have variable region here and constant region over there. And you know that the variable region is going to make antigen binding site. You know this. Yeah? So the variable region is fragmented. So the gene of variable region have multiple fragments. And from each fragment, we have multiple versions. For example, from fragment 1, we have 1A, 1B, 1C, and so on. And from fragment 2, we have fragment 2A, 2B, and so on. Fragment 3, again, multiple versions. There are multiple versions of each fragment. Different fragments and multiple versions from each fragment. Okay? All of them are in the variable part of the antibody. Clear, no question? If you do not understand, ask me, explain it to you. I can explain those things anytime you need. Okay? That's okay? Please, if you do not understand, tell me, explain it to you again. They are very important. This is another figure showing the fragments of DNA. You can see here, one fragment is here, another fragment is here, another fragment is here, and from this part to the rest of the DNA is the constant regions of the DNA. And from this fragment, we have, for example, 45 versions. From this fragment, we have 23 versions. From these fragments, we have few versions. This is for heavy chain, and this one is for light chain. Again, different fragments and different versions from each fragment. It's the same for this one. Variable region, fragment 1, 2, 3, and several versions from fragment 1, several from 2, several from 3. That is shown here in another way. Okay? And the rest of the DNA is the different constants, including constant for mu. Which class of antibody? This one we produce mu, IgM. Delta, gamma 3, gamma 1, subclasses of IgG. IgG3, IgG1, alpha 1, IgA1, gamma 2, IgG2, epsilon 1, it means Ig, it produces Ig. Again, constant region, variable region, and those fragments are called, we have 3 fragments in variable. The first fragment is named V fragment, okay? The other fragment is called D fragment, and the other one is J fragment. So we have 3 fragments of DNA in the variable region of antibody and TCR as well. And different versions of V, and different versions of D, and different versions of J are located in the variable region. We are talking about the gene, not the protein, not the antibody. We are talking about the gene that is going to produce antibody. So, I'm talking about the process by which T cell and B cell can produce millions of BCR and TCR from few genes. I'm talking about this process. How a T cell or B cell, not a T cell, how T cell or developing T cells and B cells in the bone marrow or thymus can produce millions of TCR or BCR. I'm talking about this process. So I told you we have variable region, we are talking about the genes, variable parts, constant parts, and in variable we have fragments of DNA, 3 fragments, and for each fragment we have multiple versions, and those, in We are talking about variable region of the light chain and heavy chain, that both of these variable regions make the antigen binding site of antibody. We are talking about this part only, not the constant part of the antibody. And indeed, you might remember of antibody, antigen binding site, and just I'm talking about this part. The variable region of heavy chain and the variable region of light chain. All the process happens on the gene of this part, not on the protein, not on the mRNA. I'm talking about gene, I'm talking about the DNA. Everything happens on the DNA. Not RNA, not protein. Before transcription and before translation, this process happens to produce many TCRs or BCRs. So in the light chain, we have 2 fragments, and in the heavy chain, we have 3 fragments. Fragments 1 and 2 for light chain, and 1, 2, and 3 for heavy chain. That those fragments are called F8, indeed is variable. The second fragment is called J chain or joining region, and the fragment 3 is called diversity. Yes. So 3 fragments on heavy chain, 2 fragments on light chain. So the diversity of light chain is higher or the heavy chain? Heavy chain. Because we have 3 fragments. Clear? So, multiple V regions, V versions. For example, in human, on heavy chain, we have 45 different V fragments and 23 D fragments and few J fragments. On the heavy chain, we do not have D fragments. We have just J and V. D. On the light chain, we do not have D fragments. Okay? Variable region, fragments, multiple V fragments, multiple D fragments, multiple J fragments. Now we are going to produce TCR or BCR by using of these different versions of D or combination of different versions of B with D and with J. We can produce different TCRs and BCRs. What does that? For example, when we use V2 from V fragments, D3 from D fragments, and J from J fragments, we will have this part is indeed a V region of the antibody. Okay? Another composition, V3, D1, J, another, JN for example. So we have a combination here and another combination here. Combination first and second, they are different from each other. Clear for you or not? This is called combinatorial diversity. It means we have a pool of fragments and using different fragments of V plus D and plus J, we can produce different combinations. We can produce another combination, for example, V1, D3, and J2. It's again a new fragment. This one is against one epitope, this one is against another epitope, this one is against another epitope. So, from multiple fragments and versions, we can produce so many variable regions. All of these parts make the variable region of antibody. For one chain. That's because we have 3 fragments, so they are for heavy chain. So all of them is indeed the V domain of the antibody. Is that clear? I think no. Yeah? Yes or no? If yes, please say yes. If no, please say no.
Just the last one. Last video.
Yes, this part only. Yes, I'll tell you. So, until now, I told you that we have fragments, all of these parts. You've seen that all of these parts is V, or variable region of the antibody. All of these parts. Variable region of the antibody, the gene of variable region, is composed of 3 fragments and multiple versions of each fragment. In the process of recombination, of rearrangement, we can use these fragments together to make different variable region. We can use, for example, V fragment from here, one fragment from here, another fragment from here, another from J, to make a composition, a combination, that this combination, for example here, J2, D3, V2. This combination is different from this combination because here we have J and D1, not D3. D1 is different from D3, and V3, not V2. So this is a new combination. And this new combination is indeed one V region. And this new combination is another V region. And these combinations are against different epitopes because they have different shapes. Because they have made from different fragments. Okay? Is that clear? Don't think about the exam. You are just thinking about the exam. Think about the topic. Don't worry about it. If you understand, you will answer the questions.
How will you get a question from this?
I cannot give you an example question. Finish the slides, I'll tell you one example question. I'll tell you. But don't think about the exam. Think about the concepts, not the exam. If you understand the concept, you will pass the exam. Absolutely. If you do not understand the exam, you won't pass the exam. So the understanding of the concepts is important. So,
Thinking of question is part of successful exam.
yeah, maybe. No question? Is that clear? It doesn't matter. It doesn't matter. Yes, for example, let me explain it in another way. This V region can be V1, can be bound to D1 and J1. This one is a combination. Then V1 again can bind to D2, can bind to J1, another combination. Again, V1 can bind to D3 and J2, another combination. There is no restriction. We can mix them together to make new combinations of them. If you have, for example, 3 different fragments and 3 multiple versions of each fragment, we mix them together to produce different combinations. When we say different combinations, it means we have different shapes. It means we have different variable regions. It means we have different specificity. From fusions, we can make thousands of specificity. Got it? No question? Clear? And this, when we say, for example, in this combination, we use V1, V2, D3, and J1. it means that we have to cut the DNA from this part, and those parts is eliminated. And then from this part of the DNA, we need to choose this one. Again, we need to cut the DNA. The DNA is broken several times. And then they are binding to each other here. They are not free pieces. They are bound to each other. Okay? We have cutting of DNA and binding of them together. This cutting and binding is done by some enzymes that they are called RAC1 and RAC2. They are very important because there are several types of immune deficiency diseases that people cannot produce this enzyme. There are some immune deficiency diseases that the patient cannot produce those enzymes. So there is a question for the exam. If a patient doesn't have those enzymes, what will happen to that patient? Why? When there is no enzyme, there is no cutting and binding of the fragments together. So the patient cannot produce different antibodies, cannot produce multiple specificities.
No diversity.
There is no diversity, yes. When there is no diversity, so the patient cannot fight with different antigens. So they are very, very important enzyme in the production of diversity in the TCR and BCR. And that's why they are called recombination activating genes. They do recombination, bridging of the DNA and binding together DNA. We are talking about DNA. All the process I have talked about it until now was on the DNA. The fragments are on the DNA, and cutting of them and binding them together and making different combination, all of them are done on the DNA, not mRNA, not protein. We are talking, when we say gene, we are talking about DNA. For example, the light chain is located here on chromosome. One light chain we have is on chromosome 2, another on chromosome 3. For example, heavy chain, we have the VDJ area is just this small part. Just this small part is the location for VDJ area or the immunoglobulin genes. The rest of the chromosome is for the rest of the proteins of the body. So from this small part, that a combination enables T cell and B cell to produce thousands of BCR and DCR just from this small part of the DNA. Okay? So, by having that process is called combinatorial diversity. It means production of diversity with combination of the different fragments. So it is called combinatorial diversity. Combinatorial diversity. But it's not enough. We have 45 fragments of V, 23 of, for example, there is no need to memorize the number of those fragments. Knowing the process is enough. Having few V fragments and few or several D fragments and few J fragments cannot make millions of BCR. Can make just thousands of BCR. And no, it is said that this combinatorial diversity can produce 2 or 3 million BCRs or TCRs. Just 2 or 3 millions of BCRs and TCRs can be produced by combinatorial diversity. But I told you, we need more than 10 million diversity. We need more than 10 million, or we need billions of diversity sometimes. He said, I told you sometimes we need 10 to power of 9 or even sometimes 10 to power of 10 billions of diversity. But with combinatorial diversity, just we have 2 to 3 millions of BCRs. We need another step to increase the diversity. Until now, I talked about one step that it enables T cell and T cell or BCL to produce 2 or 3 million TCR or BCR from fragments, from combination of different fragments together. But it's not enough. We need another step to complete this diversification process. Yes, that's junctional diversity that I will speak or talk about. Thank you. No question? Question, comment? So, is that clear for you that combinatorial diversification or combinatorial diversity is not enough to produce millions or billions of TCR and BCR? Do you know what's the reason behind it? Why? Let me ask the question in another way. Why does combinatorial diversity is not enough to produce 10 million or billions of different BCRs? Let me ask a question in another way. Is combinatorial diversity enough to produce millions of BCR and TCR, billions of BCR and TCR, or 10 millions of BCR and TCR, or not? It's not. Why it's not?
Maybe because the rest of fragment is eliminated.
No. We do not have enough fragments. We have just several fragments. In order to be able to produce 10 billion or 10 million or billions of TCR, we need to have more fragments. And again, we need more genes. And there is no capacity in the DNA. There is no, the number of fragments is not enough. Okay? That's why combinatorial therapy cannot produce a billion of receptors. It just can produce 2 or 3 millions. That's all. 2 or 3 millions. 2 or 3 millions is not enough. We need 10 millions. Okay? There is another process to increase the diversity of antibody or BCR or TCR, which is called junctional diversity. Because I told you, the breaking of DNA, cutting of DNA, and binding to the DNA together is made by the enzymes. Okay? When the fragments are broken, for example, V13 are broken, V2 are broken, and J4 are broken as well. Before binding of them together, in the junctional site, we can add some nucleotide or delete some others. Okay? There are some enzymes that in the junction of the fragments, between the fragments, that they are broken now, we can add some nucleotides. So this breaking, this cutting is made by RAG1 and RAG2, the enzymes needed for recombination. But there are, and they are the sequence of DNA, there are some enzymes that now they cut the DNA. Okay? They are cutting the DNA randomly. And then there are some other enzyme which is called TDT, is another enzyme, and this enzyme adds new nucleotide to the end of this one and another nucleotide here. and another nucleotide here, and another one here. So the fragments are same, but from those same fragments, by adding and deletion of some nucleotides, we will have different sequence again. For example, this part is different from this part, is different from this part, and is different from this part. Again, we have different versions of same fragments. All of them are V13, V2, and J4. But they are different from each other. Because in the binding site of the fragments, we have new nucleotides. And we have some deleted nucleotides. So the sequence is changed. As the sequence changes, we will have a new protein. It means we have a new V region. Clear? So, from one combination, B13 plus D2 plus J4, again we can produce now unlimited. There is no limitation here. Because enzymes can cut and add any nucleotide they want. There is no limitation here. It is called junctional diversity because anything happens in the junctional site of V13, the fragments, any fragment. Huh? No question? Question? Or not? Junctional diversity. And so until now, I told you that how from few genes B cell and T cell can produce millions of TCR or millions of BCR from just few genes. Two processes is done. The first one was combinatorial diversity, which indeed was the using of different fragments, combination of different fragments to produce different combinations of the fragments. And another one was junctional diversity, that means deletion or addition of new nucleotide to the binding site of the fragments together, and making new combinations or new versions of same combination. You can see here the combination is same, but the fragments that are added to the junctional site is different. And by these two processes, T cells, developing T cells and B cells can produce billions of TCR and BCR. It's amazing process in biology. It's really amazing. Producing billions of different proteins from few genes. And this process happens in both BCR and TCR. And the same process happens for both heavy chain and light chain. In the heavy chain we have 3 fragments, there is a difference. But in light chain we have just 2 fragments, V and J. There is no D fragments. But the process is same. When we have fewer fragments in the light chain, so diversity of light chain would be different from heavy chain. But one antigen binding site is composed of one light chain and one heavy chain. So one combination and junctional diversity has occurred on light chain, another one has occurred on heavy chain, and binding of them together makes the variable site or antigen binding site of the antibody. Here, same process happens for making of this part, another process in another gene happens to produce the variable region of light chain. Okay? That's okay or not? And same process happens for TCR, T cell receptor, that I'll shortly talk about. So until now, we have only talked about the variable region, antigen binding site. We haven't talked about constant region of the DNA. We haven't talked about how one B cell can produce different classes of antibody. IgM, IgA. When we talk about IgM, IgA, IgG, and the rest of the isotypes, we are talking about this part, not variable region. I told you that these isotypes have different heavy chains, that the difference is on the constant region of heavy chain. Is that clear or not? Yeah? Now, until now, I have talked about variable region. Now, I'm going to talk about the constant regions. So, until now, we have multiple or millions of V regions that now they have to bind to constant region. Mu, delta, gamma, and so on. You can see here again, now that variable region is shortened indeed as a result of the combinatorial diversity and junctional diversity. And this combination, this variable region, have to bind to one of those constant region in order to make a complete antibody. If this V region bind to this part, it will make IgM. I think I have something. For example, if this part bind to M, it will produce IgM and IgD. So, it is important to know that I've told you, in all primary response of B cell, we have just the production of which class of antibody? IgM and IgD. IgD, we do not have free IgD. IgD is mainly BCR. It's on the surface of B cell. But we have IgM both on the surface of BCR and in the circulation. We have free IgM, secreted IgM by B cell. So, in all primary immune response of B cell, we have IgM and IgD only. We do not have other classes of the antibody. I'm talking about primary immune response. When there is no memory, it's the first time of a B cell, a naive B cell, that is recognizing its specific antigen or epitope. So, if this combination bind to this part, it is called IgM. And if it is bind to this one, it is called IgD. Does the specificity changes here?
No.
Okay, again. If this combination binds to M constant region, it will produce IgM. If this combination binds to D constant region, it will produce IgD. Does the specificity of those two antibodies to IgM and IgD now is different or not?
Same.
The specificity is same because the variable region is same. When we talk about specificity, we are talking about variable region. But when we changing the constant region, we have nothing to do with the specificity or variable region. The isotype or class grouping of antibody is just because of constant region, not variable region. Again, if this constant variable region bind to G3, it will produce IgG3. If again that combination binds to G1, it will produce IgG1. If again that combination binds to E, it will produce IgE. And if the combination binds to A, it will produce IgA. So again, is the specificity of those antibodies different or not? No. The specificity is same because the variable region doesn't change between them. The only part that is changed is the constant region. So a single B cell can produce different classes of antibody with the same specificity. Yeah? I'm talking about single B cell. A single B cell can produce one specificity but with different classes. I'll talk about it. That's why we have class switching in antibody production. I told you, class switching, changing of the class of antibody. I'll talk about it. So this one is, for example, all of them are against epitope HA. It's the name of it. All of them. But this one is anti-HA IgG3, anti-HA IgG1, anti-HA IgE, IgA, and the rest of the constant regions is possible. And all of them, now we have antibody on the surface of B cell. This one is B cell. They are all B cell. Question? Comment?
Clone of B cell that have the diversity in the constant region.
Yes. When we talk about one B cell, the diversification, production of billions or millions of BCR, we are talking about the whole population of B cells, millions of clones. But when we talk about one B cell that its variable region is made, can produce, it doesn't mean diversification, it means production of different isotypes of antibody or classes of antibody. Yes, one single B cell with the same variable region, with same specificity, can produce different classes of antibody.
That have the different constant region?
Yes, the difference between these classes is the difference between constant region.
Yeah, just one B cell have different.
Just one B cell can produce, not I do not talk about the specific time. Over time, it can change the constant region. I'll talk about it, which is named class switching. I do not talk about a specific time. In one specific time, the antibodies can only produce one constant region. Except there is an exception for the primary immune response, that a B cell can produce both IgM and IgD together. In the previous slide, a single B cell with same specificity, it is a naive B cell. A naive B cell that the with same specificity can produce two classes of antibody, IgM and IgD simultaneously. Okay? All naive B cells of the body have two classes of antibody on their surface as BCR, IgM and IgD, but with the same specificity. Yeah? Clear? Yes, because the V region is same. When the V region is same, there is can recognize just one type of epitope, same epitope. Yes. The difference is in the constant region. The constant region is D here, but for this one is Ig, is M. So we have a B cell that it has two types of antibody. For example, few IgM and few IgD. These all naive B cells have two types of receptors. One receptor IgD, another IgM, but the specificity is same. Got it? How are you? difference, for example, here is different from this one. I'm talking about the sequence of the protein. This part is different, but the specificity is same. They can recognize just one epitope, but the subclasses is different and their signaling for B cell activation is different. They will produce different signals sometimes for B cell activation because the constant region is different, but they bind to same epitope. Okay? Got it? Got it, all of you?
If they bind to the same epitope, why they will have different antibody?
You are talking about naive B cell. Just on naive B cell, we have two classes. Just on naive B cell, we have IgM and IgD.
And after differentiation, we will have just one class after differentiation.
Yes. After differentiation to effector or memory, we have just one class. But on the naive B cells, we have two classes, IgM and IgD. Only on the naive B cell. And it is used in for example, in the research to discriminate naive B cell from effector B cells. On the naive B cells, we have both IgM and IgD, but on the effector, just one of them, or IgG, IgA, IgE, one of them. Okay? It is used in the laboratory, in research laboratory, to discriminate between naive and effector B cells. Clear? Yes. So, say the specificity with different constant regions. I'll talk about it again. And from one stem cell in different stages, we will have finally a mature B cell. A mature B cell have IgM and IgD simultaneously. Yeah? And this is a naive B cell. There is no need to talk about them. You can see here two antibodies, two BCRs. So B cell story. Until now, I talked about B cell development. And now I'm going to talk about B cell activation and humoral immune responses. No question, no comment? Okay. Please get back to class 15 minutes later.